Hello, and welcome to this Edison TV interview with Dr. John Reeder, founder and chief scientific officer of Sareum, a UK-based drug development company focused on novel small molecule kinase inhibitors. Welcome.
Thank you, Aaron. It's great to be here.
So, first of all, congratulations on the phase 1 data publication for SDC 1801 in the British Journal of Clinical Pharmacology. To kick off, could you start by maybe summarising some of the key takeaways from that, and then help us understand if there was sort of any other insights into that beyond the initial top-line readouts?
Yeah, sure. First of all, it was great to get the paper published in a peer-reviewed journal, so it's really giving that sort of seal of validation to the data that we've put out there. The findings were really directed in 4 different areas, and it's fairly standard for a phase 1 first-in-human study. So we're looking at safety, pharmacokinetics of a molecule, pharmacodynamic effect, so is it interacting with the targets we want it to, and we also added a food effect study into the project, so that's to see if the absorption of the drug is affected by whether you're eating prior to taking the pills or whether you're fasted when you take the capsules. So yeah, those 4 different parts. Really just to take them in order, the safety signal was excellent. We couldn't have really hoped for much better. No serious adverse events, no deaths, no real safety signals of concern at all. We look at ECGs, we look at a wide range of different levels in the laboratory, and we didn't really see any clinically significant changes in any of those, so an excellent safety signal. We did see some mild, moderate, what are called treatment emergent adverse events, but no real difference between the treated groups and the placebo groups. Just kind of things like headaches, mild myalgia, so muscle ache. But again, no real difference between the treatment groups and the placebo, so that's great. So yeah, from a safety point of view, really, really happy. The pharmacokinetics point of view, so we were getting good levels of the drug into the subjects. We had a really good half-life. It's quoted between 15 to 27 hours, I think, which is quite a wide range, but typically we're seeing half-life around about 18 hours. The point is that's suitable for once daily dosing, and that's quite important. So yeah, very happy with the pharmacokinetics. We also see a sort of quite a mild curve, not a big spiky curve. What that means is that the maximum concentration is not getting up into the area where it might affect off targets, for example. So yeah, good to see that. And then from a pharmacodynamic point of view, are we actually interacting with the targets TYK2 and JAK1 that we want to affect? Well, yeah, we generated some good evidence to suggest that we are. So we looked at, I suppose, 4 biomarkers. One's called interferon gamma-induced protein 10, IP10. Okay, it's a marker it's considered biomarker of autoimmune disorders. It's primarily driven by a JAK1 effect. So we were able to show that our molecule reduced these levels of a JAK1-driven biomarker. Likewise with another one called high sensitivity C-reactive protein. That's a general marker of inflammation. You see that rise in diseases such as cancer, autoimmune diseases, diabetes, etc. And again, we were able to show a good reduction in levels of IP10 when people were treated with our drug. And then we looked at a couple of it's ex vivo markers. So we took blood from the volunteers on the trial, challenged it with a inflammatory cytokine called interferon alpha. And then we looked at how certain proteins in the blood were affected in the presence of our drug. So we're looking particularly at phosphorylation of proteins called STAT1 and STAT3. And we were able to show that phosphorylation was reduced in a dose-responsive manner when our drug was there. So some really good evidence to show that we're inhibiting JAK1 and TYK2 with our molecule. But the kinds of effects we saw were not caused by off-target interactions with the drug. So yeah, great to get that pharmacodynamic validation of the molecule. And then the 4th thing was the food effect study. We did that so you treat patients who have just eaten a meal. Sorry, not patients, volunteers. These are healthy volunteers, and that's something I need to stress. These are not patients, healthy volunteers, but they have a meal. You treat them with the drug. Or they're fasted, you treat them with the drug, and you're looking for any differences in the exposure of the drug in those different conditions. And we didn't really see anything. There was a marginal increase of exposure in the people who'd had a breakfast, but it was not really significant. And that's great. So that gives you maximum flexibility in a dosing schedule for people when we move into clinical trials and patients start to take this. We're not saying you've got to take this with you, with a meal. You
Sure.
take it at your convenience, really. So yeah, good results all round. And then also, I think the other thing we found was we used 3 different capsule formats, and the top dose, which was a 50 mg capsule, didn't really release as much drug as we'd hoped it would. So we've needed to, since we got this data, we've been working on improving the formulation so that we can deliver the high doses of SDC 1801, which from a safety point of view, we could deliver, but we need to do that with a sensible number of capsules. So yeah, that's just something we've been working on since we got the data.
Excellent. So sounds like a highly comprehensive publication. We saw as well there were some comparisons between 1801 and other TYK2 JAK inhibitors. So it'd be great to hear in your own words what you think differentiates your lead candidates.
Yeah, so I mean, I think the lead molecule in this space is a molecule called brepocitinib. It was developed at Pfizer, but it's being developed currently by a company called Priovant. And I mean, they've recently completed a phase 3 trial in dermatomyositis. So it's likely that they'll be applying for marketing authorization later this year or early next year. So that's really the trailblazer in this area. And what we found with our molecule was, so we could compare it very nicely with some data that Pfizer, as it was at the time, published around brepocitinib in their phase 1 trial, where they did it's called the multiple ascending dose, the MAD phase of the clinical trial. They did a 10-day study, just as we did, and they were able to dose at 100 milligrams, and they started to see some really serious adverse effects. So these were reductions in certain blood components, reticulocytes and neutrophils, and they also see an increase in a protein called serum creatinine, or creatinine. It's found in the serum, okay, and that's a marker of kidney damage. Now, I don't think brepocitinib causes kidney damage, but what it does is inhibit a transporter in the kidneys. And Pfizer put out some good evidence to support that. But it's a problem because it is a marker of kidney damage. And if you take that forward, primary care physicians, for example, might see this and start to worry. Okay, so it's just something to avoid if you can. So we were really happy to see that at an equivalent exposure in our MAD study, we did not see any of these effects. We didn't see effects on hematology, and we certainly didn't see the serum creatinine effect that we think is dose limiting for brepocitinib.
Sareum recommenced phase 2 enabling tox studies earlier this year. It'd be great to hear a little bit more about how the program has progressed since then as you move towards phase 2.
I mean, I can't really go into details on that, but just to say that those studies are ongoing. Mm-hmm. I mean, I think I can probably say they're nearing completion.
Okay.
Okay. So yeah, we're optimistic about the outcome of those studies, but it's something that really we're going to have to talk about in the future, as the results become available, the data becomes available, and we will update our shareholders accordingly. So that's one to watch out for, really. I think the other thing we've been doing that I sort of alluded to is this formulation optimization. So in order to get the highest exposure in cohort D of the MAD study, we had to dose the subjects with 140 milligrams, but we had to give that as 10 milligram capsules. So that was 14 capsules. We went to a twice daily dosing. So they were having 7 capsules on each dosing event. That's not really sustainable. It's not something you could take forward into a phase 2 clinical trial, certainly.
Okay.
So the other thing we've really been focusing on is improving that formulation so that we can deliver the necessary dose of SDC 1801 in a sensible number of capsules, okay? And we've made really good progress on that. I think we're very close to what is called locking the formulation. So basically saying, this is what we're going to work with in the future. We're very close to that. And then we have to get those capsules on a stability study. This is to make sure that, they've got a reasonable shelf life. And those studies will sort of go on over the latter half of this calendar year. So yeah, that's where we're at the moment. And this is really to deliver this phase 2 ready package, which we've said we'll have the data for by the end of calendar year 26.
Yeah, well, we certainly look forward to following the progress. Taking a step back now, TYK2 JAK inhibitors, they've shown potential across a whole host of autoimmune indications. So for Sareum, does psoriasis remain the lead sort of target indication, or it'd be great to hear if there's any other sort of opportunities you think could be applicable to the candidate?
Yeah, well, I think one of the arguments we make about a dual TYK2 JAK1 inhibitor is its potential in a wide variety of autoimmune diseases. So, I mean, there are selective TYK2 molecules on the market and soon to be on the market. They've shown some good efficacy in psoriasis. I think the thing with psoriasis is it's an extremely crowded space, very competitive. There are biological therapies that are available and show really good results. There's the selective TYK2 molecules which are showing good results, and there are newer ones coming along behind TYK2 that are improvements on TYK2. And then there are some other oral peptide therapeutics. There's a molecule called icotide which has really shown pretty good results in psoriasis. So it's great. The outcome for psoriasis patients over the last few years has really moved on. It's a very crowded space. I think SDC 1801 could be competitive in that area. And I think certainly if Sareum were to take the project forward, there's a lot of advantages in doing a psoriasis trial. Okay, you can access the patients relatively easily. There are a lot of them.
Sure.
You can there are really well-established protocols in place and endpoints, so you know what you're looking for. There's a very low placebo effect. That's important because sometimes some of the autoimmune diseases do suffer from a high placebo effect and can really confound your data. So there are some definite positives. And then I think the other thing is that brepocitinib, as I mentioned, has been studied in psoriasis. So there's comparative data that we could directly compare SDC 1801 to the result. It's a caveat, different patients, different centres, so it is, you have to be careful comparing across clinical trials, but there's some data we can directly compare to. So psoriasis is great. However, I mean, brepocitinib has shown good efficacy in a number of diseases now. Alopecia areata, I mentioned dermatomyositis earlier. There's an eye disorder called uveitis. It's demonstrated good efficacy in that. So that's really sort of enabling us to look at other areas and see where the strengths of this dual TYK2 JAK1 approach lie. I think we're looking for partners for the project, and we're really hoping to identify someone who can explore these different therapeutic areas. But if we can't find a partner or strike a deal that's beneficial to us, I think psoriasis is a place we could move forward in. Yeah.
Just before we wrap up today, could you summarize for our audience the key milestones and potential catalysts to watch out for across the next, say, 12 to 18 months?
Yeah. So I mean, we've just completed our financial year end, so obviously we'll be getting our numbers and our annual report out. They're usually late September, early October sort of time. So that's sort of business as usual, I suppose. Then it's really the outcome of the ongoing toxicology trials that I've mentioned. It'll be progress in the formulation and the stability studies. I'm not saying we'll specifically announce results from these via RNS, but we'll keep our shareholders informed as much as we're able to. And then obviously we have ongoing partnering discussions, not just around SDC 1801, but also our other leading candidates, the SRA 737, the CHEK1 inhibitor, and SDC 1802, another dual TYK2 JAK1 kinase, but positioned more in the oncology space. And then we've got the collaboration with Receptor AI that we announced mid last year, where we're looking for CNS penetrant, brain penetrant TYK2 JAK1 inhibitors as well. So we're making some interesting progress in that area, and we'll hope to be announcing some data around that program as well over the course of the next year or so.
Fantastic. Lots to look forward to then.
Indeed. Well, hopefully.
Fantastic. John, thanks very much for joining us today.
It's been a pleasure. Thank you.